What if I told you that the world's first synthetic drug, a chemical so powerful it was used to fight malaria, stain cells under microscopes, and even treat poisoning in emergency rooms, has been sitting in plain sight for over 130 years? And yet, almost no one talks about it. This is the story of methylene blue, a brilliant blue dye born in the golden age of German chemistry, tested on the battlefields of 2 world wars, approved by the FDA, and studied in some of the most prestigious labs on Earth. It has saved countless lives. It remains on the World Health Organization's list of essential medicines to this day. But despite all of this, methylene blue has been largely forgotten by modern medicine, not because it stopped working, but because the story of how drugs get remembered and how they get erased is far more complicated than most people realize. This is Medicine Lost Vault, and today, we're opening the file on one of the oldest, most versatile, and most overlooked pharmaceuticals in human history. To understand methylene blue, we have to go back to 1876. The place is Germany. The Second Industrial Revolution is in full swing. Factories are belching smoke, railroads are expanding, and a quiet revolution is happening in chemistry labs across the country. Germany in the late 1800s was the undisputed capital of synthetic chemistry. Companies like BASF, Bayer, and Hoechst weren't yet the pharmaceutical giants we know today. They were dye companies. The textile industry was booming, and whoever could create the most vivid, most stable synthetic dyes stood to make a fortune. It was in this world that a chemist named Heinrich Caro, working at BASF, first synthesized methylene blue in 1876. It was a beautiful, deep blue compound, and it was immediately useful for dyeing textiles. But its story was just beginning. Just a few years after its creation, methylene blue caught the attention of a young medical researcher who would go on to become one of the most important figures in the history of medicine. His name was Paul Ehrlich. Ehrlich was fascinated by a simple but revolutionary idea. He noticed that certain dyes would stain specific types of cells while leaving others completely untouched. If a dye could selectively bind to a particular cell, he reasoned, then maybe a chemical could be designed to selectively target and destroy disease-causing organisms while leaving healthy cells alone. He called this concept the magic bullet. And methylene blue was one of the first chemicals he tested. In 1891, Ehrlich used methylene blue to treat 2 patients suffering from malaria. The results were remarkable. The blue dye appeared to target the malaria parasites in the blood. This was a landmark moment, one of the earliest examples of a synthetic chemical being used to treat an infectious disease. Paul Ehrlich would later win the Nobel Prize in 1908, and his magic bullet concept would become the philosophical foundation of modern pharmacology. But the drug that helped inspire it all? It would take a very different path. Methylene blue's role in fighting malaria didn't end in Ehrlich's lab. It followed soldiers into 2 world wars. During World War I, malaria was a devastating problem for troops stationed in tropical regions. Quinine, the standard treatment derived from the bark of the cinchona tree, was in dangerously short supply. Germany in particular was cut off from quinine supplies by Allied blockades. So German military scientists turned to methylene blue as an anti-malarial alternative. It worked. Soldiers were given the blue compound, and it helped reduce malaria infections. But there was a notable side effect that would become legendary. It turned the soldiers' urine bright blue. This cosmetic quirk, while medically harmless, created significant resistance among troops who found it alarming. By World War II, newer antimalarial drugs like chloroquine had been developed, partly inspired by the chemical framework that methylene blue had established. Methylene blue was gradually phased out of frontline malaria treatment, not because it failed, but because newer options were available. Still, methylene blue had proven something crucial. A synthetic dye could fight disease. That idea changed medicine forever. While methylene blue was being tested on battlefields, it was simultaneously revolutionizing another field entirely: laboratory science. Remember Paul Ehrlich's observation that methylene blue could selectively stain certain cells. That property made it one of the most important tools in the history of microscopy. In the late 1800s, scientists were racing to understand the invisible world of bacteria and cells. But there was a problem. Most cells and microorganisms are nearly transparent under a microscope. You couldn't see them clearly enough to study them. Methylene Blue solved this. When applied to tissue samples, it would bind to specific structures, making them visible under magnification. It became essential for identifying bacteria, studying nerve cells, and mapping tissue structures. Robert Koch, the legendary microbiologist who identified the bacteria responsible for tuberculosis and cholera, used methylene blue staining in his groundbreaking work. Without it, some of the most important discoveries in infectious disease might have been delayed by years. To this day, methylene blue staining remains a standard technique taught in biology and medical labs around the world. It is one of the most enduring contributions of this remarkable compound. Now here's where the story takes an important turn. Because methylene blue isn't just a historical curiosity. It's an FDA-approved drug that is actively used in hospitals right now. Its primary approved indication is for a condition called Methemoglobinemia. This is a potentially life-threatening condition where the hemoglobin in your blood becomes chemically altered and loses its ability to carry oxygen effectively. Your blood essentially stops doing its job. Methemoglobinemia can be caused by exposure to certain chemicals, drug reactions, or in rare cases, genetic conditions. When it happens, the patient can turn a disturbing shade of blue as their tissues become starved of oxygen. Without treatment, it can be fatal. Methylene blue is the frontline treatment. When injected intravenously, it acts as an electron carrier, restoring hemoglobin to its functional form. It literally reverses the condition. It works quickly, it's effective, and it has been the standard treatment for decades. This is not alternative medicine. This is not a fringe therapy. This is textbook emergency medicine, approved by the FDA, used in hospitals worldwide, and listed on the World Health Organization's list of essential medicines. Methemoglobinemia treatment is the headline, but it's far from the only established medical application. Methylene blue has been used as a surgical dye and diagnostic tool for decades. Surgeons use it to visualize anatomical structures during operations. It helps identify fistulas, map sentinel lymph nodes, and trace urinary tract connections. Its vivid blue color makes it an invaluable visual aid in the operating room. It has also been used to treat a condition called ifosfamide-induced encephalopathy. Ifosfamide is a chemotherapy drug that can sometimes cause serious neurological side effects. Methylene blue has been documented in medical literature as an effective treatment for this complication. Additionally, methylene blue has a long history of use as an antiseptic, particularly for urinary tract infections. Before the age of modern antibiotics, it was one of the few tools doctors had to fight bacterial infections of the urinary system. Each of these applications has been documented in peer-reviewed medical literature. Each represents a legitimate, recognized use of this compound. And yet, if you ask most people, even many doctors, to list what methylene blue is used for, most would struggle to name even one. Now we come to what might be the most fascinating chapter in the methylene blue story. And I want to be very clear here, because this is where we move from established medicine into active research. What I'm about to describe are findings from published, peer-reviewed studies, not proven treatments, not medical advice, but scientific investigations that are ongoing. Starting in the early 2000s, researchers began studying methylene blue's effects on the brain. What they found caught the attention of the neuroscience community. Studies published in journals including the Proceedings of the National Academy of Sciences and various neuroscience publications found that low doses of methylene blue appeared to enhance mitochondrial function in brain cells. Mitochondria, as you may know, are the energy-producing structures inside every cell. In the brain, mitochondrial health is critical. Research teams at institutions including the University of Texas at San Antonio published studies suggesting that methylene blue could improve memory consolidation in animal models. Rats given low doses performed better on memory tasks. Brain imaging studies in small human trials showed increased activity in regions associated with attention and memory. These are published findings in legitimate scientific journals. They are intriguing, but they are preliminary, and much more research is needed before any clinical applications could be established. Perhaps the most closely watched area of methylene blue research involves Alzheimer's disease. And again, I want to emphasize that this is active, ongoing research, not an established treatment. Alzheimer's disease is associated with the accumulation of two types of abnormal protein structures in the brain: amyloid plaques and tau tangles. For decades, most pharmaceutical research focused on amyloid plaques. Billions of dollars were spent developing drugs to target amyloid. Many of those drugs failed in clinical trials. Meanwhile, a researcher named Claude Wishick at the University of Aberdeen in Scotland had been studying the other protein, tau, since the 1980s. Wishick's research led him to methylene blue. His lab published studies suggesting that methylene blue could inhibit the aggregation of tau proteins, potentially slowing one of the key processes involved in Alzheimer's progression. This led to the development of a modified form of methylene blue called LMTM, or leukomethylthioneinium. Clinical trials were conducted. The results have been mixed. Some trials showed potential benefits in certain patient subgroups, while others did not meet their primary endpoints. The research continues. The scientific community remains divided on the significance of these findings. What is not in dispute is that methylene blue represents one of the few compounds that has been seriously investigated for tau-related approaches to Alzheimer's. Whether it will ultimately prove effective remains an open scientific question. So here we are. A compound that was the world's first synthetic drug, that helped pioneer the concept of targeted medicine, that fought malaria in 2 world wars, that remains FDA-approved and on the WHO essential medicines list, that is being actively studied in neuroscience labs around the world. Why doesn't anyone talk about it? The answer lies not in the science, but in the economics and structure of modern pharmaceutical development. And this is a pattern we'll see again and again on this channel. Methylene blue was first synthesized in 1876. Its patents expired well over a century ago. It is a generic compound that can be manufactured for pennies. No pharmaceutical company can obtain exclusive patent protection on it. And this is the key issue. The modern drug development pipeline is built around patents. A company invests hundreds of millions of dollars in clinical trials with the expectation that a successful drug will be protected by patents for years, allowing them to recoup their investment and generate profit. Without patent protection, there's no financial incentive to fund the massive clinical trials needed to get new approvals. This doesn't mean anyone is hiding methylene blue. It means the economic system that drives drug development doesn't have a mechanism to fund large-scale trials for compounds that can't be patented. It's a structural problem, not a conspiracy.
Let's look at this more closely, because it's important to understand the mechanics. The average cost of bringing a new drug to market is estimated at over $1 billion when you factor in the cost of failed trials. Phase 3 clinical trials alone, the large-scale studies needed for FDA approval, can cost hundreds of millions. For a patented drug, a company can expect roughly 10 to 15 years of market exclusivity. During that time, they can price the drug to recover their investment. It's an imperfect system, but it's the engine that drives most pharmaceutical innovation. Now consider methylene blue. Any company that invested in large-scale clinical trials would bear all the costs. But the moment the drug was approved for a new use, any generic manufacturer could produce it. The company that funded the research would have no way to recoup its investment. This is sometimes called the generic paradox. The drugs that have been around the longest, that have the most safety data, and that are cheapest to produce, are often the least likely to be studied for new applications. It's not because they lack potential, it's because the business model doesn't support it. Methylene blue is one of the most striking examples of this paradox. But it's far from the only one. In recent years, methylene blue has experienced something of a quiet resurgence in certain medical communities. But this resurgence has come with its own set of complications. Some physicians have begun prescribing compounded forms of methylene blue for various off-label uses. Compounding pharmacies can prepare customized formulations of approved drugs, which is a legitimate and regulated practice. However, the quality and consistency of compounded preparations can vary significantly. The FDA has expressed concerns about the purity and safety of some compounded methylene blue products, particularly those sold online without proper medical oversight. This is an important distinction. Pharmaceutical-grade methylene blue, manufactured under strict FDA guidelines, is a well-characterized drug with a known safety profile. Products of uncertain origin and purity are a different matter entirely. This situation highlights another challenge facing older generic drugs. Without a major pharmaceutical company overseeing quality control and distribution, the market can become fragmented, leading to inconsistencies that can undermine both safety and the compound's scientific reputation. No honest examination of any drug would be complete without discussing its limitations and safety concerns. Methylene Blue is no exception. While Methylene Blue has a generally well-established safety profile at approved doses, it is not without risks. It can interact dangerously with certain medications, most notably serotonergic drugs, which include many common antidepressants. The combination can potentially trigger a serious condition called serotonin syndrome. The FDA issued a safety communication about this interaction in 2016. At high doses, Methylene Blue can actually cause the very condition it's used to treat, Methemoglobinemia, through a paradoxical effect. It can also cause nausea, headache, and other side effects. In patients with a genetic condition called G6PD deficiency, Methylene Blue can trigger a dangerous breakdown of red blood cells. These are not minor considerations. They underscore why Methylene Blue, like any pharmaceutical, should only be used under proper medical supervision. The fact that it's old, cheap, and available does not mean it's safe for unsupervised use. Anyone interested in Methylene Blue research should discuss it with their healthcare provider. This video is educational content about the history and science of a fascinating compound. It is not medical advice. Despite the economic challenges, Methylene Blue research hasn't stopped. It's actually accelerating in some areas, largely thanks to academic institutions and government-funded research programs that aren't dependent on patent-driven returns. The National Institutes of Health has funded multiple studies investigating Methylene Blue. Research continues at universities across the globe, from the United States to Europe to Asia. Published studies have explored its potential in areas ranging from traumatic brain injury to septic shock to antimicrobial resistance. One particularly active area involves Methylene Blue's role as a photosensitizer. When activated by certain wavelengths of light, Methylene Blue can generate reactive oxygen species that destroy microorganisms. This property has been studied for antimicrobial applications, including as a potential tool against drug-resistant bacteria, a growing global health concern. Published research in these areas continues to appear in peer-reviewed journals. Whether any of these investigations will lead to new clinical applications remains to be seen. Science moves slowly, and the gap between a promising lab finding and a proven treatment is vast. But the research pipeline is active. The scientific community has not forgotten Methylene Blue, even if the broader public has. Methylene Blue is not unique. It's part of a broader pattern in pharmaceutical history that affects dozens of older compounds. Throughout the 20th century, many drugs were developed, used successfully, and then gradually pushed aside as newer, patented alternatives arrived. Some of those newer drugs were genuinely superior. Others were primarily different enough to be patentable, even if their clinical advantages were modest. This isn't a conspiracy theory. It's a well-documented feature of pharmaceutical economics that has been discussed in medical journals, policy papers, and government reports. Organizations like the National Institutes of Health and the World Health Organization have acknowledged the challenge of studying generic compounds. Several potential solutions have been proposed. Extended exclusivity periods for companies that conduct new research on old drugs. Government-funded clinical trials for promising generic compounds. International research consortiums that share the costs and benefits of studying unpatented drugs. Progress has been slow. The economic incentives are deeply entrenched. But the conversation is happening, and awareness is growing. This is why channels like this one exist. Not to tell you what to take or what to avoid, but to make sure these stories don't disappear entirely. Let's take a step back and appreciate the remarkable timeline of this single compound. 1876. Heinrich Caro synthesizes Methylene Blue at BASF as a textile dye. 1891. Paul Ehrlich uses it to treat malaria patients, pioneering the concept of targeted chemical therapy. Early 1900s. It becomes a critical tool in microscopy and biological staining, helping identify the bacteria behind tuberculosis and other diseases. World War I. German military uses it as an antimalarial when quinine supplies are cut off. World War II era. Newer antimalarials inspired by Methylene Blue's chemical framework begin to replace it. Mid-20th century. It becomes the standard treatment for methemoglobinemia, and finds uses in surgical visualization. 1980s onward, researchers begin investigating its effects on tau proteins and neurodegenerative diseases. 2000s to present, studies on mitochondrial function, memory, and neuroprotection appear in peer-reviewed journals. Clinical trials for Alzheimer's-related applications are conducted. Today, it remains FDA-approved, listed on the WHO Essential Medicines list, and actively studied in labs around the world. 150 years, one compound, a story that spans the entire arc of modern medicine.
Methylene Blue isn't a miracle drug. It isn't a hidden cure for anything. What it is one of the most remarkable and enduring compounds in the history of pharmacology, a chemical that helped birth the very concept of modern drug therapy, that has saved lives for over a century, and that continues to reveal new scientific possibilities in research labs today. The fact that most people have never heard of it tells us something important, not about methylene blue itself, but about the systems that determine which drugs get attention and which ones fade into obscurity. On this channel, we go into the vault. We pull out the files that time has buried, not to promote miracle cures or fuel conspiracy theories, but to tell the full story of medicine, including the chapters that got left out. If this is your first time here, consider subscribing. We release new investigations regularly, and every single one explores a compound, a treatment, or a medical story that deserves to be remembered. I'm Medicine Lost Vault, and this is just the beginning. Disclaimer: this video is for educational and informational purposes only. It is not medical advice. Always consult a qualified healthcare professional before making any decisions about your health. The research discussed in this video includes ongoing investigations that have not yet resulted in approved treatments.